Hi everyone, welcome back to the next video. We'll continue our discussion with this surprising observation from the LTE that landscapes may not have peaks and adaptation may continue forever. Let's just look at where we were. So, we have this time versus fitness data. This is the point of 20K, this is the point of 40,000 generations, and using experimental data of fitness with the first 20,000 generations, I built two models. Both these models made excellent predictions of the experimental data. This was one, and the second model maybe what this is not good. The second model predicted something like this, and both these models were made with the data that was collected for the first 20,000 generations, and after that these models were asked to predict what would happen in the next 20,000 generations. This is the prediction that was made by model 2, and this is the prediction that was made by model 1. And when we actually compute the fitnesses of the next 20,000 generations, what we saw was that these fitness values in the subsequent 20,000 generations are much better predicted by model 2 as compared to model 1. Remember, model 2 was of the form that y is equal to at plus 1 to the power b. We call these a dash and b dash depending on their values determined by the data from the first 20,000 generations, and model 1 was y is equal to 1 plus a naught t plus b naught by t. And what these results clearly show that this model is a better predictor. Now, if that is the case, then the question is that this model also says that there is no fitness peak, because fitness will keep on increasing. There is no Fmax associated with fitness on a landscape. So, there is no fitness peak. In this one, if I plug t equal to infinity, y also will reach the value infinity, which means that fitness will keep on increasing forever, and that seems very counterintuitive. Eventually, when it comes to cell division, there are laws of physics that have to be respected, and cell division is a process that does need finite amount of time, and cells cannot divide into two infinitely fast. So, how do we think about the fact that this model is doing a better job at prediction as compared to this one? And I think there are two reasons to not be sort of dismayed at the fact that fitness does not have peak and that model is a better predictor than the other one. And the first reason is that the prediction that this model is making is better in a very short term, is better in the time window of 20,000 to 40,000 only. We do not know, evolutionary change takes place, large evolutionary change takes place over millions of years, and we do not know what is going to happen if we were to ask these two models to predict what would happen in the next 1 million generations. So, to illustrate this idea, let us imagine this, and now let us change the scale that this is 20,000 and this is 40,000. And we start with a fitness of 1, and model 1 predicts something like this. And of course, there is a F max here, which model 1 will not cross. So, this is F max. On the other hand, the second model tells us a slightly different story. It also starts with 1, but maybe its trajectory is 40,000 in 40. Maybe its trajectory was something like this. Let me draw the model 1 again, and I get something like this. So, maybe over a very long period of time, the model predictions look like this. This is always capped by F max, which it will not cross. So, what we have seen so far with the experimental data from LTE is the fact that both these models do a great job at prediction in 20,000, but when it comes to 40,000, model 2 becomes a better predictor. This is model 2. But over a really long time scale, maybe the fitness values as we move forward in time will begin to look something like this. And hence, when I look at this, let us say this is 500,000 generations. If I look over evolutionary time scale, really long time scales, then I would still find that the reality is between model 1 and model 2, and model 1 is sort of a better predictor as compared to model 2. So, just because the model which said that there is no fitness peak was a better predictor in the window of 20,000 to 40,000 generations does not mean that it will continue to be a better predictor as we move forward in time. So, a scenario like this might play out. So, we can summarize this by saying that model 2 is a better predictor between 20K and 40K generations. This does not imply that it is going to be a better predictor in all time. That is the first reason I think that why it should not be so hard for us to believe that model 2 is a better predictor here. That is sort of the first reason. The second reason is a commentary on this facet that Lenski wrote on the website that is dedicated to LTE. And remember that while model 2 says that there is no peak, while model 2 implies that there is no peak, the rate at which fitness is increasing, rate of increase of fitness is actually continuously slowing down in model 2. In model 2. So, the time required for fitness to increase by a certain amount is going to get increasingly longer as we move towards higher number of generations. Fitness is increasing, but that rate is slowing down all the time. And we have this fitness related to this model, which is a dash t plus 1 to the power b dash in model 2. And what this model, what the values of a dash and b dash show, that in 1 billion generations, if I were to plug in t as 1 billion generations, remember t is time measured in number of generations, the y that I get, the fitness that I will get as a predictor from model 2 is that the E. coli after 1 generations, although who knows if it will still be E. coli after 1 billion generations, E. coli or the bacterial species that we get after 1 billion generations will divide every, I think it is of the order of every 5 minutes. So, while 5 minutes is extremely rapid, we know of bacteria that divide in times which are close to that time. Is it inconceivable that in a time which is as long as 1 billion generations, the adaptation that is taking place in those flasks will not evolve these organisms to divide every 5 minutes? To my mind, this prediction that this model makes is not impossible. So, while the idea that no fitness peak and hence infinite fitnesses seems a little ridiculous and counterintuitive, I think these two mitigating factors sort of take care of what model 2 is trying to tell us. And although I will not be around to see what happens in 1 billion generations, but I would not be surprised if E. coli was dividing every 5 minutes or 6 minutes or of that, something of that nature once 1 billion years, 1 billion generations of this experiment have been processed. All right. So, that is the discussion of this paper. This paper was published in 2013 in the journal Science. Next, we come to perhaps what is considered the most interesting observation from LTE, and this was something that happened in the 2000s. So, let us go back to the experiment, and we have 12 identical flasks . These are 12 identical lines which are being evolved in identical environments which were seeded by an identical E. coli strain, and so on and so forth. So, everything about them has been precisely the same ever since the experiment was started in 1988. The culture media in which they are growing has this characteristic that we are trying to study their adaptation. We are trying to study that how bacteria cope with low amounts of glucose as the only carbon source that is available to them. So, low amounts of glucose as the carbon source in the environment. Spelling is wrong, low amounts of glucose. Now, however, at the start of experiment in the 80s, what was also done was that this media was supplemented by citrate in the flasks. Citrate was added in this culture media when the experiment was started. And remember that the conditions in which these bacteria and flasks are being propagated forward is 37 degrees Celsius, shaking, which means that these bacteria that are growing have access to oxygen. And this, while it was not realized at the time, led to the most surprising observations from this experiment. E. coli has this characteristic property that In anaerobic conditions, when oxygen is not around, it can utilize citrate as a carbon source, but in oxic conditions, when there is oxygen around, E. coli cannot use citrate as a carbon source. That is a characteristic property of E. coli which has also been used historically to perform diagnostics on whether a pathogen or a bacterial isolate is E. coli or not, that it cannot use citrate in aerobic conditions. Despite that, and obviously at the start of this was known at the start of the experiment, and despite that, citrate was added to the culture media when the experiment was started. The sort of the purpose behind adding citrate was that it chelates metal ions better. So, it makes historically citrate has been added to culture media to make these growth curves more uniform and more consistent and not depend so much on the quality of water and other reagents that are being added to the media. So, that is the premise of adding citrate to these flasks. But what that also leads us to is the fact that there is this lots of resource available to bacteria which is not being currently used. The experiment is being done in this environment. However, if the environment was this, then E. coli can use this resource for their growth. But imagine a bacteria in the flask where everybody is fighting for the small amounts of glucose present. If I as an individual pick up a mutation which allows me to utilize citrate for carbon source, that would be a great beneficial mutation because that will give me unfiltered access to this giant pool of reserve that is present in the culture conditions. So, I could get access to it, but obviously there would need to be mutations which need to happen before I can start to utilize citrate. But what I would like you to realize is that in this flask, there is some amount of glucose. Let us say this is glucose and there is lots of citrate. This is citrate and the bacteria can only utilize glucose, and now if one of these bacteria picks up a mutation that allows it to use citrate for growth also, then this mutation would be a great mutation because it's allowing it unfiltered access to this great resource which is not being used by anybody else in the population. However, this was not anticipated that that such a mutation will happen, and there was no basis for us to think that such a mutation can take place and such an evolutionary change will result from this experiment. So, what has happened since start of the experiment is that we have these 12 lines of these experiments. One of these lines at around generation 31,000. So, in order to understand this, you have to understand the logistics of how an experiment like this is conducted. Since subculture from one flask to another flask happens every 24 hours, so what this would mean, and the same happens in my lab for instance, is that since this subculture of 1:100 is happening every day at the same time, let us say this transfer happens at 10 a.m. This is full of bacteria and we are going to transfer. So, at 10 a.m. somebody needs to come to the lab, take 1:100 fraction of this and transfer it to a new flask which contains the same recipe of nutrients available to it. And because this is happening every day, you develop a familiarity of what a flask looks like when you come in after 24 hours of growth. So, flasks which have media which do not support a lot of growth will have some bacteria in it, and these bacteria will manifest as this haziness in the culture. As more and more bacteria grow, if you have a flask which contains lots of resources, then more bacteria will be there, and those cultures after 24 hours of growth will look much more denser. So, if you are a microbiology student who's done this kind of thing, you develop an intuition for how dense a culture is every day, and you expect that it has to be the same density today also. However, in the course of this experiment, what happened at some point around this many number of generations is that 11 flasks on one day, when somebody came to transfer, 11 flasks had the technical term is optical density, and this is just a measure of how densely the culture had grown in this flask. So, in 11 flasks the optical density was normal as usual, but in one flask the density of the culture was much higher. So, as someone who performs evolution experiments, when you see something like this happen, your first instinct is to sort of is to say that what must have happened is that some contaminant bacteria must have gotten access to the flask, either during the process of transfer or something must have gone wrong, and you had this contaminant come into this particular flask. As a result, this culture is just totally overcrowded because of this contaminant growth. Because the bacteria that I am working with does not grow to such high densities. I know that from prior experience of every single day when I come in the morning to do these transfers. Hence, there must have been something else that happened, and the most common cause of that something else is that some contamination happened in the growth media. So, that's sort of the first explanation and the most common and obvious explanation when something like this happens. So, this culture, this flask was checked for contaminants. However, it was found that there was no contamination, and the flask, none was found, and the flask only contained bacteria from the experiment. And then further analysis revealed, so if that is the case, that means that why is the number of bacteria gone up so much? Because these bacteria are growing only on glucose as the carbon source, and there is only a small amount of glucose as carbon source. So, glucose can't support the carbon requirements of so many cells that would support such a high density. And then it was found that actually this flask's density has gone up because this has cells which are growing on citrate. In the presence of oxygen, which E. coli does not do. So, that is what we have as the most surprising observation from LTE. This happened soon after the turn of the century, a few years after 2000. So, what is going on here? Now, even after 80,000 generations, we only have one line which is growing on citrate. None of these 11 lines have learnt to use citrate in this time. None of these 11 lines have citrate utilizers. So what does that tell us? That obviously that some mutation happened which facilitated utilization of citrate in this environment. But what sort of a mutation was that only one line in all this time, remember this happened at around 30,000 generations. Another 50,000 generations have since passed, but citrate utilization as a capacity has not yet evolved in any of the other 11 lines. So what does that tell us? What could be the reason that only 1 out of 12 lines has done it in all this while? And again, the fitness advantage to be had if I as an individual in one of those flasks evolve the capacity to utilize and grow on citrate, the advantage that I have compared to other bacteria in the population is huge. So why hasn't this ability evolved in other flasks? So this could be because of citrate. The ability to utilize citrate could have evolved because of two reasons. The first possible reasons, two possible reasons. The first possible reason says that a very fortuitous lucky mutation happened, a very lucky or a rare mutation happened in flask I, in flask of line I, which facilitated utilization of citrate. Now that makes sense because maybe it required duplication of certain parts of the genome followed by a deletion, followed by a SNP. So all these events needed to happen, and maybe this was a very precise and a very lucky mutation that needed to happen. And because suppose this is the genome of the individual, maybe you needed just this exact piece of DNA maybe to get flipped, maybe you needed this region to get flipped, and only then citrate utilization would evolve. Any more or any less of the region being flipped wouldn't be able to do it, and hence that kind of event happens only very, very rarely, and this was a very rare such mutation that happened in this line. It could have, that rare mutation could have happened anywhere else. Alternatively, line I acquired mutations in the time leading up to acquired mutations leading up to 30,000 such that individuals in this line were primed to use citrate. These mutations happened which brought the cell to a point where it was just ready to utilize citrate, and one more mutation and then it is ready to utilize citrate. such that one additional acquisition, one additional mutation would lead to citrate utilization. These prior mutations may have conferred benefit in terms of being able to grow better on glucose, but one of the side effects of acquisition of these prior mutations was that if one of the cells in this population acquired this one precise mutation, a regular one, not a rare mutation, if this one happened, then it is ready to utilize citrate. If this same mutation happened in any other line, but because its prior mutations are other mutations, this would not be able to use citrate, because this mutation only confers the ability to utilize citrate when it happens in conjunction with these precise mutations. So, this mutation itself was not lucky, but one of these lines needed to have these prior mutations such that this mutation can happen and then subsequent utilization of citrate could happen. So, these two are alternative hypotheses, and we will discuss which one of these two hypotheses was found to be right and what would be the experiment that you do to check which of these two hypotheses was the actual one leading to citrate utilization. We will continue that in the next video.